Hello and welcome to today's lecture, where I will be talking about phase field modeling of active matter. What I'm going to do is to discuss how we use this phase field modeling to understand interface properties in active material. And I will build heavily on many of the things that you have already learned in the previous lectures, especially on active fluid dynamics and Lattice-Boltzmann methods, and also active nematics. And what I'm going to do here is that I'm going to describe this phase field modeling in the context of the research problems that we are looking at. So the details of the phase field model and the derivation and the fundamental features of it is included in the lecture notes, and I would recommend that you read the lecture notes carefully. Here, I want to give it context in terms of how we are using this phase field modeling in understanding properties of active materials. In particular, I will be discussing these in the context of two of the most important implications of active matter theory and why we are using active matter. The first category is that we use active matter to understand fundamental questions about the physics of living systems. For example, we now know in cancer invasion, at least in humans, that when cancer invades, in more than 90% of the times, it does it collectively. And when it does it collectively, we can see that we are dealing with an interface between an advancing active matter, here cell colony, and the surrounding that we need to capture. Another beautiful example is during organ development. So this is a fly embryo at the early stages of the formation. And what you would see is after a while, all these cells, they coordinate their motions all together and they form these beautiful stripy patterns on the surface of the embryo. Each of these stripes will then go on and form different parts of the fly body. And again, we use understandings of active matter in order to understand this complex pattern formation on the surface of the that is forming this fly embryo. But in addition to providing this understanding of physical systems, we are also actively using these studies of active matter in order to mitigate, in order to mimic nature, to make our own materials that are capable of doing some of these functions that biological systems are able to do. And this is a subject of intense research. Many studies are going on using these fundamental understandings from active matter to design new types of materials. For example, I will discuss these living vesicles later on, and people have even looked at using this motion, collective motion of bacteria to power microscopic gears and building micro machines that are capable of producing mechanical work. So what I'm going to do today is that I'm going to discuss two examples from the first category where we use physics of active matter, and in this case, phase field modeling to understand actually biology of living systems. And then I will discuss two examples of how we use these understanding for the design and control of active material. So the first example I want to discuss concerns with bacteria. And what we are dealing with is a highly infectious bacteria called Pseudomonas. And these Pseudomonas bacteria, they normally propel themselves by grabbing on surfaces. They have these small feet called pili, and they use these pili to grab on the surface and to move. What our experimental collaborators do is that they make a mutant of these bacteria that has more legs. And when it has more legs and you check it under microscope, you measure the velocity, you see that it moves 2 times faster. So we have the wild type, which is the normal Pseudomonas infectious bacteria, and then we make a mutant of this infectious bacteria that moves 2 times faster. Now we ask if this faster moving, this individually moving faster, gives any advantage in terms of collective invasion. And to do that, we culture the bacteria in a racetrack. So we have here this wild type, individually slow moving, and on the bottom we have the fast moving one. What happens is quite amazing. As the race begins, initially the fast one takes over, but after a while, the slow one is actually the one that is winning the race. Why is that happening? So we wanted to understand why this sort of counterintuitive behavior emerges collectively when bacteria invade spaces. We first asked, so maybe there's something with the racetrack geometry. Does the same thing happen when we mix these 2 types of bacteria together? And then we have here basically the mixture of the fast moving that is shown by purple, and the slow moving ones are the gray bacteria. And we see the same behavior in a mixture, that as the time goes by, the slow moving bacteria actually dominates the front of the invading colony, while the fast moving ones actually they get stuck in some structures that we term rosettes. So if we understand why these rosettes form, then we would have understanding of why these fast-moving bacteria are not able to expand and invade the new territory as effective as their slow-moving counterpart. To understand why these rosettes form, we look more carefully at the colonies. And first thing that we notice is that when we look at the orientation field of these bacteria, they show behaviors very similar to nematic liquid crystals. And they show these 2 types of nematic topological defects, the plus half that has this comet shape and the minus half that has a trefoil shape. And I'm sure that you have learned about this in the active nematics lecture, so I will not go into more details of what these defects are. The important thing is that we observe these half-integer topological defects, which implicates that these bacterial colony they are behaving as nematic systems. But they are not any nematic system, they are active. And that is very clear when you look at the dynamics of these defects. These defects get constantly created and then they also annihilate each other. First step to understand this, we make the most simple possible model that we can think of. We consider a bunch of rods that are interacting with each other through volume exclusion. So there's no attraction, there's only repulsion when to avoid any overlap between the rods. And each rod has a force on it to give it self-propulsion. And as such, the dynamics is basically governed by simple Langevin dynamics. Qualitatively, we observe the same thing. We get this constant creation and annihilation of topological defects. But more importantly, we can compare the velocity field around the topological defects from this very simple model and the actual experiments. And as you can see, it actually matches quite nicely and shows one fundamental properties and distinctions of the plus half or the comet-shaped defect from the trefoil defect. And that is the plus half defect has this broken symmetry, and as such, it is basically self-propelled. It moves towards its head direction. Next, we ask if the continuum theory would produce the same result. And this is basically the continuum active nematics theory that I'm pretty sure you have been already introduced to. And just a quick recap, this consists of considering the nematic tensor that relaxes to the minimum of free energy that is described by the Landau-de Gennes expansion plus the Frank free energy. And in addition to that, we have the co-rotation term, because the elongated particles, in addition to moving with the flow, they also should respond to gradients in the flow. If there's vorticity, they should rotate. If there's shear, they should align with the shear. And this is coupled, of course, to the mass and momentum conservation, where the momentum conservation has this extra bit, which is the active stress that you have also learned. So we consider this theory. And the advantage here is that we can just consider the form of the topological defect. We know the director field for that. So we can impose Q. And then with the imposed Q, we know the active stress, and we can analytically solve for the velocity using Green's function. We do that, we get analytic description of the flow field, and that matches perfectly with what discrete model, and more importantly, the experiments predict. And again, the important point here is that the plus half defects are self-propelled, as you can see here. And the other important point is that from the theory, we know if the bacteria are more active, we expect these plus half defects to be faster. And that's exactly what they see in the experiments when they measure the mean square displacements of defects. You can see that the minus half defects, they are the same between the slow and fast moving bacteria, while the plus half defects move much faster in the fast moving bacteria. At this point, then, so maybe we understand what's happening. In the fast-moving bacteria, the defects are moving faster. So the fast-moving defects then are able to come closer to each other, and they essentially form a virtual full integer defect that has this shape of the rosette that I discussed. What happens at the full integer defect is that it has a lot of energy, so it is not sustainable, and what happens is that the bacteria escape to the third dimension. And we believe that is what's happening in terms of the fast-moving bacteria, that they create these fast-moving defects that form the rosette, so they stand up, and as such they get stuck, so they cannot invade as effective as slow-moving. Indeed, we can check it in the simple rod model, and we can see when we shoot two defects to each other, for the slow-moving ones, things more or less stay in place. For the fast-moving ones, they basically stand up, and they form this kind of reverse domino effect. More importantly, the same thing happens in the experiments, and it's exactly at these points that the two defects come together, the bacteria get out of focus. That means that they stand up, and the same thing happens also in the mixture. So this simple example shows why this study of active matter is important, in a sense of giving us ideas about the biological functionality of the topological defects. This in the bacteria that seems to have put some kind of fundamental speed limit on their motion, and give sort of evolutionary advantage to the wild type slow moving bacteria. Now, if we consider this bacteria colony and the way that it invades and expands in the environment, we need to basically capture, in addition to the active pneumatic behavior that the bacteria colony shows, we need to also capture the interaction with the surrounding environment. As you can see, if bacteria starts dividing and starts moving, then you have this expanding colony that essentially infiltrates the surrounding. In order to capture this, we consider the equations of the pneumatic liquid crystal, the active pneumatic liquid crystal, but in addition to the existing equations that I quickly reviewed earlier, we also consider additional order parameter called phase field order parameters. And the idea is that I want this phase field, I call it phi, to distinguish these two regions. The region inside, when you have this active pneumatic behavior, I indicate that by phi equal 1 by design. And I want, on the outside, I want this parameter to take value of 0. So now if I have some evolution for this phi order parameter that is coupled to this pneumatic behavior, then I can distinguish the active region from the passive region. That's the idea. So This is the equation that we had previously. Now notice that in the free energy, I have included this coupling to phi. What this means is that if phi is 0, this term basically drops off. And if I minimize this free energy, this part of the free energy that determines the relaxation to the minimum of the free energy, I will get 0 order. While if phi is 1, this will be the regular relaxation free energy, and it will give me nematic order. And if I have nematic order, then I have active state. So the equation that I add for phi basically consists of the advection and, again, the relaxation to the minimum of the free energy as explained in the lecture notes. This here is essentially chemical potential that separates the two regions from each other. And if I have an expanding colony, I can have a source term in phi here that would basically represent the cell division and the activity that is caused by the cell division and results in the expansion of the colony. Let's see how this works. So this was the bacterial colony that was expanding. And now, if I use this phase field modeling to look at the expanding colony of active nematics, I notice that essentially once the defects formed, here shown by the plus half defects are shown by the circles, the defects move towards the interface. And this movement towards the interface basically changes the shape of the colony. If you look more carefully, you will see essentially the same thing in the experiments. As the bacteria divide and they move, you see that these protrusions essentially form at the locations of the plus half defects. As you can see here, here, and the minus half defects basically get produced in the center. So at this point then, we had the idea that this defect motion basically mediates the morphology of the growing colony, as you can see here. And one thing that, for example, we checked was that by increasing the friction, when you increase the number of topological defects, if you increase the friction of these active bacteria with the substrate, then you get more topological defects and the shape of the colony becomes more isotropic because there are more defects that are moving around to the interface and you get more circular shape compared to the original case where you have the few number of defects, they move in different directions and then you have sort of anisotropic geometry. And what determines this interaction of defects with the interface is basically, again, the velocity field that these topological defects create. They create these large vortices, and essentially if you look at the correlation between the defect and the interface velocity, that we see that there is a well-defined length scale that also coincides with the vorticity length scale that is being produced by these defects. Another interesting property that we see in these active interfaces is that if you look more closely at this bacterial colony where you have the topological defects, you would also notice that at the interface, the bacteria are all aligned parallel to the interface. This is a phenomenon called active anchoring, and it is a direct consequence of the active stress generation by these bacteria that we have shown analytically in the course notes, and you can also measure it in the simulations, and you can see there is this tendency of anchoring parallel to the interface, which is the direct consequence of the activity of the particles. Now, I'll switch gears, as I promised, to discuss the second example where we can use these understandings of active matter system for biological problems. And this is now looking at a completely different system, which is the epithelial cells. These are the cells lining surfaces and cavities in our body. And what we found in collaboration with experiments is that, again, these plus half defects and minus half defects form in epithelial colonies, meaning that they show this active nematic behavior. But more importantly, at the location of these plus half defects, cells die and they get expelled from a 2D monolayer. Not only we found it for kidney cells, then they checked the experiments also for human skin cells, for aggressive and non-aggressive breast cancer cells. Seems like this is some kind of universal behavior. And these are essentially, if you are looking at a cell in a tissue or a cell monolayer, these white spots are the points where the cells get extruded out of the monolayer. Is extremely important in order to maintain the integrity of the tissue, because cells are constantly dividing and the tissue needs to also get rid of some of the cells. And the example of this, you can very clearly see that if we map the elongation of the cells as the nematic director, then we can see at the location of the topological defects that form, you have these white spots where the cells are being extruded. Again, we wanted to understand why this happens at the location of +half defects. So again, if you compare the active nematic theory with the experiments, you get a good agreement in terms of flow fields, but more importantly, in the experiments, they can measure the stress field, the mechanical stress that these cells exert under substrate. And if you measure the isotropic stress, which means how much tension or compression there is, one can clearly see that at the head portion of +half defects, there is this wide blue region, wide negative stress. So it means that there's high compression that essentially pushes the cells out. And this you can see also in the experiments, that before the extrusion, this negative stress builds up. So we reformulated how cells get extruded out of the colony. We know that the cell monolayer is an active system. It forms ±half topological defects. They move around. And at the head portion of these defects, the large compression is essentially enough to kill the cell and almost simultaneously push it out of the monolayer. Now, the important question here is that these monolayers, they have one fundamental difference from the bacterial system that I discussed earlier, and that is, for the cell monolayers, the shape is a dynamic variable. It is not like a bacteria that has a rod-shaped configuration. For cells, the shape, in many cases, is just isotropic, and it constantly changes as these cells push and pull on each other. So we wanted to understand how this gives rise to this formation of the topological defects and active behavior in this system. And to understand this, then we use a different formulation for phase field modeling. Now here we are thinking as phase field for modeling the interface between individual cells. So each cell is therefore represented by a phase field order parameter, and the interaction of these many phase fields gives rise to the tissue behavior. So essentially, now compared to the previous case, I have phi I, where I basically represents the index for each cell. If I have 100 cells, I have hundreds of these phase fields. And for each phase field, phi equal 1 would demark the interior of the cell, and phi equal 0 will demark the exterior of the cell. As before, we have the relaxation to the minimum of the free energy that determines the cell properties, as I will explain. And here we have the velocity field that basically is determined again from the force balance condition. Now, so the passive part, which is this free energy, as I mentioned, defines the properties of the interface, as is for the phase field models. And it consists of the Canhilliard free energy. This would be essentially, this would determine the relaxation of the free energy towards the minimum. You would have a double well potential with phi equals 0 and phi equal 1 as minimum, and would have the penalty for the gradients that would give some kind of a diffuse interface. In addition to that, we can introduce an area constraint, meaning that we want the cells to have a certain area. So it would penalize deviation from this pi r squared area. And we can also introduce additional terms that would determine the repulsion between different phase fields or adhesion between different phase fields. But additionally to this free energy dynamics, we also have the active dynamics that is set by this velocity field. And the velocity field essentially is determined from the force balance. So all we need to describe is basically the forces that are acting on the cells. And to describe the forces, we can use the stress description for the tissue. And the simplest possible stress description that we would consider is an active stress that is proportional to the shape tensor for each cell. It's very similar to the active nematic stress that you were introduced before, but instead of defining a nematic tensor, now we have a shape tensor for each cell, which its largest eigenvalue and would determine the order, and the largest eigenvector would determine the direction over which the cell has elongated. And the active stress is proportional to this shape, meaning that if there is a little elongation, there will be some kind of a bootstrap effect that would basically reinforce itself and would elongate the cell further. And that would be the source of activity that would then elongate the cells that are in contact with this cell, and that would drive the dynamics. This is the most simple way of considering the activity in the phase field model. And it is basically also motivated from this kind of experimental force measurements that shows that cells basically exert this kind of dipolar force fields along the direction of elongation that drives their motion to have a collective dynamic behavior in a monolayer. So if we consider this kind of phase field, then we can see that indeed this sort of active stress proportional to the shape tensor can drive the collective motion. Here, each cell is shown, the interface of the cells is shown, and the lines in each cell, they show basically the director field determining the elongation of the cells. And there is certainly a threshold in the activity that needs to be passed in order to create this collective motion. And this threshold also increases with cell stiffness. The stiffer the cell, then you need larger forces in order to deform the cells and to create these motions that we observe. But more importantly, using this model, we are able to also capture the stress fields around the topological defect. So again, the stress field from this phase field model matches the continuum model and matches the experiments, which is promising in terms of using this kind of modeling in order to describe dynamic behavior in cellular monolayers. So again, this highlights another example of using active matter understanding, and in particular phase field modeling, to find some kind of biological functionality and to understand some kind of biological process. And I hope by this point you are convinced about the importance of active matter, and in particular active nematics, and more specifically topological defects in terms of biological systems. And there is also growing evidence on the role of these topological defects in different realizations of biological systems, for example, in human fibrosarcoma or in neuroprogenitor cells, where cells accumulate that plus have defects. And the same thing happens for the myoblast cells, that these defects have the functionalities in terms of determining the collective behavior. Now, these defects are important, but one problem is that when you look at these, for example, human bronchial cells and their dynamics of topological defects, or swimming bacteria, the defects have these sort of chaotic dynamics. This is what you probably have already been introduced to in the previous lectures as the active turbulence, which is accompanied by the chaotic motion of defects. Now, if these defects are important and they have biological functionalities, then it is also demanding that we find now ways that we can control the behavior of active matter systems, such that instead of showing these chaotic behavior, then we can have some form of control over the patterns that they create. Again, this sort of chaotic pattern is known as active turbulence. That is on its own, it's a very fascinating topic of research. I'm just including a number of references that you can read more about it. But here I'm going to just focus on, again, two examples of how we can use active matter simulations, and in particular, phase field modeling, in order to have some ways of controlling these chaotic patterns of active matter motion. So the first strategy is using some kind of pattern medium. And this is based on experiments from Oleg Lavrantovich's lab in Kent State, where the brilliant idea is to disperse these bacteria in a background passive liquid crystal. The advantage is that we can control the orientation of passive liquid crystal. And by doing that, we actually have control over the orientation of bacteria. They are free to swim, but their orientation we have control over. And what we did in this work was to design patterns that have this C shape, meaning that you have these bends, you have patterns of bends here, and in between the bends, you have these splay patterns. You introduce the bacteria to the system, and what happens is that bacteria, they get focused, they get depleted from the bend regions, and they get focused in these splay regions. And they form very nice unidirectional strong jets in these regions of splay, as you can see in this video here. These jets, they get more and more bacteria attracted to them, and after a while, the active pneumatic instability kicks in. However, the instability cannot grow. Instead, it gets basically confined by these bend patterns that are on the top. So you can form these nice oscillatory jets that consists of many bacteria and are moving relatively fast. And of course, then if you have those kind of jets, then you can use it for example, transporting microscopic cargoes. And here I just show examples of considering microscale colloids, single or multiple colloids, both in the jet and also in the oscillatory patterns that are being transported in real time using these bacterial streams. Now, to model this system, we basically consider what I explained before in terms of the continuum active pneumatics representation that is coupled to the phase field order parameter. But now, in addition to this, we introduce additional free energy term that all it says is that the pneumatic director in the regions where phi is equal 1, it wants to align to the background director, which is the passive pattern that we impose. How does it look like? So for example, we have this red director that shows the background passive liquid crystal that has these C shapes of the bend and splay. And then we have the active pneumatic phase that interacts with this background phase. And what happens if you start from a band, you can see that it basically condensates into these splay regions in agreement with what they see in the experiments. And if you measure the concentration, the profile also matches quite nicely the experimental pattern. And more importantly, can capture the instability and the amplitude of these oscillations, and can see that they cannot grow over the amplitude that is set by the bend patterns in the system. As you increase the activity, initially you have no instability, you would have a perfect jet. And after a while, you form the instability, but this instability cannot grow larger than the amplitude set by the bend patterns. This is a very nice example of using the phase field model to look at the interface between a moving active phase of matter and a background that has some kind of pattern into it. The other example in terms of control of active matter, which will be my last example of these lectures, is using topological constraints. And this is motivated by really nice experiments when they consider taking these microtubule motor protein emulsions. I'm pretty sure you have been introduced to these systems as one of the popular realizations of active matter systems, which shows the topological defects. And what they did in these experiments in Andreas Bauch's group was that taking these microtubule motor protein mixtures and then make an emulsion of it into a droplet. And now what happens is that you have a droplet that is active. So at the surface of this droplet, there are these microtubule motor protein mixtures. They constantly move, exert stresses, and they create these fascinating patterns of motion. They constant shape deformations and also these arms and that come out of this pattern. And you can imagine that modeling such a thing is a nightmare because all sorts of complications are going in. You have the two-phase flow in 3D, and then you have these active stresses that are only acting at the surface, at the interface, and you have hydrodynamics on the background, and you have a shape that is dynamically evolving. Again, you can use phase field modeling to also attack this problem, and the idea is also quite simple. So we take the equations that we have for pneumatic and for the velocity field as before, introduce the phase field model as before, and the free energy terms are similar. You have the terms coming from the surface tension and the relaxation. We can also introduce some kind of membrane free energy in the form of Helfrich free energy that is included here. You have the pneumatic elastic energy and then the coupling between the pneumatic order and the binary order parameter as before. But the trick here is that we want the active material to leave at the interface. Previously, we were doing the coupling such that one phase was active, then there was one phase that was inactive, and in between, there was an interface. Now, we do the coupling such that only at the interface, we have the active material. And by doing so, then you can actually get patterns like this. You can have a three-dimensional, self-evolving, active matter shell that creates these very nice protrusions. You can see even -half, +half defects that are forming on this surface. Again, you can see this +half defect moving around, creating this protrusion. And then as it moves on, forms a +1 defect and leaves a -half defect behind. And indeed, we can make a zoo of shapes based on the strength of the activity and changing it from extensile to contractile. And more importantly, we can see that essentially these protrusions again form when two +half defects come together. So I have come back to where I started, now in a different context, showing that at the shell, these two +half defects coming together, they form a protrusion that moves up, and then in 3D, that would essentially form a +1 defect and its tip leaving -half defect behind, which would then show correlations between defects and the curvature on the surface. What's amazing is that these kind of similar behavior has been now recently probed in experiments in vivo. And this is considering experiments on an animal hydra. This is sea animal, and it's called hydra because it has this ability that if you cut any fragment of this animal, it would grow to the shape of the original animal. You can see an example here. Time here shows hours, and then you can see this segment that they had taken. After around 2 days, it basically deforms to the original shape of the animal, which is a fascinating phenomenon. and it shows this morphogenesis of this animal. But more importantly, in recent work, Kinneret Karen's group, they have mapped the orientation of these actin filaments on the surface of the animal and essentially find that these tentacles, they form at the positions of +½ defects, and then they move on and form +1. Mouse and foot also form at +1, and the -½ defects are left behind, as we discussed earlier. So this, again, is very promising to observe these topological defects and the predictions from the phase field modeling in real in vivo experiments, and also gives us some hopes in terms of designing this mimicking nature in order to design self-deforming material. And I would just end by saying that seems like this idea of the self-deforming material is not completely new, and maybe 30 years ago, people have already thought about it, as you can see here. What they call the liquid matter. But of course, it was a science fiction movie, but I think we have now come closer to actually at least simulating this kind of material that are able to deform their shapes and have some kind of self-organized pattern. So with that, thank you very much for listening to this lecture, and I look forward to discussing any questions that you would have in our panel section later on. Have a good day.